Well, good afternoon, everyone, wherever you are, and welcome to our webinar today. The circulatory system. What a marvel breakthrough in biological technology. If we were to take all of the veins and arteries and capillaries and stretch them out in a line, in a child, that line would be 60,000 miles long. And 100,000 miles long in an adult. That would be enough length to circle the equator 4 times. And it would only take one red blood cell 60 seconds to make a complete circuit of that system. So in essence, we have a series of connecting tubes that allows the blood to flow as a liquid. Well, yes, the blood is a liquid, but it also has a lot of elements in its content. If we were to take one drop of blood, we would see that there would be 10,000 white blood cells, 250,000 platelets, 5 million red blood cells, and in the other half of the drop would be plasma proteins. So when we put a vascular access device into the venous or arterial system, we are essentially putting a tube within a tube. 150 years ago, Dr. Virchow determined the postulates as what would create venous thrombosis. And putting a vascular access device into the circulatory system violates two of those postulates, being venous stasis and vessel injury. And in the face of hypercoagulability, even further increases the risk of thrombosis. The other problem that vascular access devices create is a portal of entry for microorganisms. Thus, we have a vascular access device that has 2 major potential complications, thrombosis and infection. Now, what types of VADs are used in the U.S.? Well, peripheral IVs, 160 million are approximately sold per year. In short-term central venous catheters, 4 million. Long-term surgically implanted catheters, 0.3 million. And PICCs are fast catching up to short-term CVCs at 2 million, and interestingly, arterial catheters more than those two combined at 6 million, but yet who's doing surveillance on those? And all of these are thrombogenic surfaces, and indeed, all of these provide a surface for colonization, so we really should be monitoring all of them. Now, the question becomes, are PICCs at a greater risk for thrombosis and infection than other CVCs? Because we're constantly battling which one of these should be used. And of course, many hospitals have committed a great amount of resources towards the PICC catheter teams. So, Pickware et al. Conducted a study where they did a systematic review of 12 studies. In each of these studies, they compared complication rates of the PICC catheter to the central venous catheter. They looked at primarily 4 complications: malposition, thrombophlebitis, catheter dysfunction, and catheter infection. Now, you'll notice that they measured these, indicating the risk for these complications. For malposition, it was a 3.8 times greater risk for malposition using a PICC catheter than a CVC. For thrombophlebitis, a 5.8 times more chance of thrombophlebitis with a PICC than a central line. And for catheter dysfunction, 6 times greater risk for dysfunction with a PICC than a CVC. With the infection, although they said that the definitions were not the same and comparative in many of these studies, but the number that they determined was 0.8, which means that there was no difference between the infection rates of PICCs and CVCs. When they combined all of the data together, they found that a statistically significant difference between the 2 types of catheters, and the overall risk of complication was 2 times greater with a PICC than a CVC. Now, this is probably not such good news. And the question becomes, should we stop using them? I say no, because they are very valuable tools in our toolkit for the care of patients. However, should we be concerned? And the answer is an emphatic yes. we need to be concerned both about infection and thrombosis. These devices are in the hands, in the domain of nurses, and so therefore we are obligated to begin to have a better understanding about the pathophysiology of these complications and indeed begin to look for new strategies as to how to prevent them. Now, we said infection and thrombosis or thrombotic events are very critical in these devices. So we're going to talk about both of them. We're going to begin with the infection component. When we have a tube, we have two surfaces, the internal surface and the external surface. And we said that these devices are portals of entry. Well, the major source of bacteria for the extraluminal surface colonization is actually the insertion site, where the intraluminal component, the major source or entry point of bacteria, is the catheter hub itself. Now, the question becomes, which surface is the highest risk for infection? Well, we finally have some insight into that question. This was a study done by the Pennsylvania Patient Safety Advisory Committee and published in September of 2011. Their purpose was to determine whether an institution should focus resources on a specific phase of CVC life to prevent CRBSI. In other words, should they be putting resources on the insertion component or on the maintenance component? And as you know, all of the campaigns and all the bundles to date have primarily been focused on insertion. So to answer this question, they did an analysis of the NHSN database for the state of Pennsylvania for the year 2010 to determine the date of infection event from the time of insertion. The sample size, or the number of infections that occurred during this year, was 653 CLABSIs. Now, here we see a graph where they plotted out each one of these infections. And you see that this goes out to 60 days, and this represents 98% of that population. So if we look at the mean time to infection, that was 14 days. The median time, or the time at which half occurred before and half occurred above, that was 10 days. But the mode, which is the day of which most infections occurred, was on day 5. This implicates that prior or up to day 5 are primarily extraluminal source of infection, and those after 5 days are primarily intraluminal infections. So the other issue here is that oftentimes in doing our surveillance, we don't really see patients, particularly for nurses who insert PICCs. They see them on insertion and don't see them thereafter. Or even for infection preventionists who may be doing surveillance only in the intensive care units, or even if they're doing whole hospital, don't see these patients after they leave the hospital. But this data and their conclusion demonstrated that maintenance is the phase in which CLABSI most likely is developed. Now, how do CRBSIs happen during the maintenance phase? Well, we all know that it requires bacteria and microorganisms to cause an infection. So somehow those microorganisms enter through a portal of entry. And we've just described those two portals as the intraluminal surface and the extraluminal. Now, when those bacteria enter through those points, then if they touch the surface of the external lumen or internal lumen, they will attach to that surface. And here you see a picture of bacteria attached to the intraluminal surface of a central venous catheter. This is what we've always called colonization, the presence of organisms in the absence of disease. But within minutes, these bacteria will begin to form a biofilm. When the biofilm completes its growth and goes through maturity, the next phase in the life cycle of biofilm formation is biofilm dispersal. This is a survival mode for bacteria in these colonies. But again, from survival, they're going to disperse bacteria to create colonies elsewhere. This is how bacteria have survived on this earth for 5 billion years. Now, back to the CVC. As these bacteria are dispersed from the biofilms, depending on how much biofilm you have, and the inoculation, if you will, that's released from them in the face of a susceptible host, if the inoculum is greater than what they can handle, then you will have infection. Now, what is biofilm? Biofilm is microorganisms. It's not clot, it's not fibrin, it's made by microorganisms who live in a self-organized cooperative community. They are attached to surfaces, interfaces, or each other. They are embedded in a self-produced matrix of extracellular polymeric substances, and these bacteria within the biofilm will exhibit an altered phenotype with respect to growth rate and gene transcription. Now here is a device, and here is biofilm on this device, and you can see that it looks very slimy and very lubricious, but you actually cannot see biofilm bacteria within this material. Now, this is probably the best picture that I could show you of biofilm in real time. And this is exactly what it looks like on a catheter hub, on the internal lumen surface, and the external lumen surface. But this is not a catheter. Can you guess what this device is? Well, time is up. This is actually a child's pacifier. And again, they're showing the large amount of biofilm that was formed on this device. So the clinical implications then are that the bacteria in the biofilms will resist all host defenses. They will rapidly promote antimicrobial resistance. They are recalcitrant to antimicrobial therapy at therapeutic doses, and they are very tenaciously adherent to surfaces. This means that in order to remove or kill biofilm bacteria, it will require both chemical as well as mechanical friction. Now, how does this apply to the internal lumen of a catheter, and how does it actually become colonized? Well, again, we're back to the portals of entry, and this is why this is such a difficult problem for the interluminal surface, because we have so many portals of entry. We have stopcocks, and quite frankly, these are very high rate of colonization, and we don't even have any recommendations for the care and maintenance of stopcocks in the CDC guidelines or in the SHEA document. So we tend to forget them, but they are very important sources. We also have injection ports, any injection port along the entire intravenous system. Needleless connectors, of course, we are familiar with, and very importantly, we've got to begin to understand and focus on the catheter hub itself as a major source of biofilm, as this is where the bacteria first enter into the catheter system. Now, the question is, how do bacteria survive on these surfaces? Well, let's come to the laboratory with me and let's do a little experiment. We have, I took some needleless connectors out of their sterile package and inoculated them with Staph aureus. And you see the Staph aureus there on the surface. But let's get a little closer. After 30 minutes, let's see what they're doing. And here you can see one set here, that cell that's actually in replication, but all of the other cells on the surface are also in replication. If we go over to the right side here, we'll see that these bacteria have already started to attach to one another to the surface and form a biofilm. Now you'll notice that these cells look like they're a little bit fuzzy, and that's because they have already started to secrete this matrix of slimy material. In SEM, of course, you have to freeze dry the sample, so this is dehydrated. Otherwise, it would look very much like it looked on the pacifier. But now what about the clinical setting? And do these biofilms actually form on patients' connectors? So I did go out to the USC LAC hospital in Los Angeles when I was at the Medical Biofilm Center, and I retrieved some connectors from patients. So this is one example of a connector, and here under the SEM, you see the plastic rim of this connector, and it looks like it has a lot of dried material, but we don't really know what that is. So let's pick a spot here, and we're going to zoom in with the microscope. And as you can see, we have, unfortunately, thousands and millions of cells, gram-positive organisms that have formed a very thick and extensive biofilm on the surface of this connector. I also took the connectors and cut the tops off, and then this particular one had a flow tube inside, so I pulled the flow tube out, cut it in half and opened it, and then scanned under the microscope. And here you see not only that the flow tube is completely lined with a contiguous biofilm, but we also have clumps of biofilm within the flow tube. And we're going to take a look here at one in particular and zoom in and see what kind of organisms we find. Well, this was actually, I'm going to call it a microbial zoo because it actually found representative from just about all species. Here's a gram-positive group. Over here to the right in the top, there's some gram-negative organisms there. These filamentous organisms resemble an hyphae or actinomyces, and we see one down here on the right is actually a Treponema, which is a normal colonizer of the oral cavity. So we've got all kinds of microorganisms that have gotten in through the connector septum. Now, how do they get there? Well, we can either have loss of integrity of the connector septum, but probably the primary way is we inject them. And it's a simple concept. Most of these connectors will have bacteria on them just by being in the patient's environment, next to their skin, next to endotracheal tubes, and so forth. So if we do not scrub them and disinfect them and remove the bacteria from the surfaces of these access devices, then when we inject, we will inject the bacteria. And it's irregardless of whether it's with a syringe or with it's an administration tubing. And the one here on the bottom can be our intravenous sets that are used intermittently. If they're put up over the IV stand and they're not protected, and we go to access our devices, then we literally put them in and inject the bacteria into the patient. Now, as these bacteria are flowing through the system, if they touch any area on the inside of the system, whether it's the connector, the hub, or the catheter, they will stick to it. And when we discontinue the flow, or even during flow, they will form a biofilm. As we mentioned earlier, when these biofilms mature, which doesn't take long, within hours to days, they will leave the biofilm and be planktonic organisms within the system. And then when we inject, whether it be by bolus injection or infusion, again, we now inject bacteria into the patient. So when they attach to the surfaces of the catheter, they can live in two forms. Either here on the left, as you see, they're directly attached to the catheter surface and are forming a biofilm, or they can attach to the plasma proteins that have settled out on the catheter surface. On the right, even if they form directly to the catheter surface itself, with subsequent blood draws, you may have a coating of plasma protein right over top of the biofilm, and they will use that as so-called housing material. That affords extra protection for the bacteria in the biofilm. Now that's the intraluminal surface. So how about the extraluminal surface? How does that become colonized? Well, one thing we tend to forget is that when we place a vascular access device, we are putting it through three tissue spaces: the skin, the subcutaneous space, and the intravascular space. And there are opportunity for infection to occur or transfer through any one of these tissue spaces. So in order to understand that, let's begin with inserting a catheter. And the first step in the procedure, of course, is to do skin antisepsis. And when you pick up the needle to place that catheter, then you're going through skin with or without bacteria at the surface. Now, how many are there at the time of venipuncture are really up to you. And it depends on how well you do or do not do the antiseptic preparation. 80% of the resident organisms live on the surface of the skin, and those or within the first 5 layers of the stratum corneum. Those are easily removed by skin scrubbing, but the other 20% live in the sebaceous glands and the hair follicles. Those are virtually impossible to get. So as we begin, I recommend doing a clean procedure with scrubbing of the skin and then the application of the final antiseptic. So here we go. There are, again, we're going to do the venipuncture, and regardless of how well you do or do not do the procedure, you will pick up some bacteria as it goes through the lower layers. So those bacteria will go in with the needle. You then place the guide wire. The next step is remove the needle and then advance the dilator and the sheath. And of course, now you're putting this through the skin, and bacteria will go into the sub-Q space along with this device. We then remove the guide wire, and then we remove the vessel dilator, and our next step will be to place the catheter. Now, I want you to think for a moment, up until now, how many times have you touched the skin with your gloves? Well, probably quite a few times. And each time you do that, again, depending on how many bacteria you left there, you will be contaminating your gloves, particularly the fingertips. The next step in the procedure is to advance the catheter. And you will be picking up the tip of the catheter as the first step. So if your gloves are contaminated, you will be inoculating the tip of the catheter, and then as you advance it and you touch the catheter, you will inoculate on each step of the way. We then remove the peel-away sheath, and we're left with the catheter in place and having taken some bacteria with you. Now, what's happening in these tissue spaces? Well, we just talked a lot about on the skin. Even if you have eradicated or removed most of those bacteria, the bacteria from the sebaceous glands and hair follicles will come to the surface of the skin and recolonize it. So we have bacteria there. And in the subcutaneous tissue, okay, you will have the bacteria that are attached to the catheter surface will form a biofilm, but you can also have biofilm forming in the subcutaneous tissue. And in this picture, you can see a colorized photo of biofilm in a wound. Now, what is the response to the catheter in the bloodstream? Well, we know that we advance the catheter, and you may already have some bacteria attached. However, within seconds or milliseconds, plasma proteins in the blood will attach to the catheter surface itself and form what we call a conditioning layer. The layer is about 100 nanometers thick and is composed of primarily fibrinogen or fibronectin. Those plasma proteins then will coat or cover up any of the biofilm that has started to form or the bacteria that have attached to the surface on the way in. Within minutes to hours, the fibrin will attract platelets. The platelets will release thrombogenics, and we have the initiation of the coagulation cascade, and a fibrin sheath will form. In the subcutaneous tissue, within minutes to hours, we will have the inflammatory response because you have created a wound. This involves mast cells release of histamine, capillary dilatation, we have plasma leaking, and followed by fibroblast invasion, granulation tissue, and finally collagen encapsulation. Now here is a picture of a jugular vein of a sheep that has been end block dissected, meaning that the catheter was removed and the skin, subcutaneous tissue, and vein were all taken out in one section. You can see the catheter there to the left, and I have excised the subcu tissue in the vein to expose the sheath that is around the catheter in the subcutaneous tissue. And then you see where the dotted line is really the entry point into the vein, and that collagen capsule continues and is contiguous with the fibrin sheath around the catheter. Now, if we were to do a cross-section of the subcutaneous tissue, we can see that collagen capsule. And in the space within, between the collagen capsule and the catheter is where your drainage will appear. Now, this goes all the way to the surface at the skin. So if there are bacteria at the insertion site, those bacteria can be transferred and dispersed through that entire column, collagen capsule in the subcu tissue and on into the fibrin sheath and into the bloodstream. Now here's a picture extending that photo and showing you the length of the fibrin sheath and that those bacteria could be dispersed here at the end of the fibrin sheath. Now, within days to weeks of the catheter being in place, you can have red blood cell attachment and a pericatheter thrombosis can form. But this is different. This is more like a thrombosis or a clot as opposed to the proteinaceous conditioning layer. Now, as far as the biofilm that has been developing now for days from those bacteria that were attached on insertion, those will mature and they will be dispersed, from the biofilm. And again, depending on the inoculum, can create a bloodstream infection. Now, if the catheter remains in for more than about 7 days, we will see a transformation of the fibrin sheath where smooth muscle cells will actually migrate from the vessel wall at the site of venipuncture and will cover the fibrin sheath. This then will be covered by endothelium that migrates again from the vessel wall. And now we will have a more collagenous type of material similar to what we saw in the subcutaneous space. What's important to note here is that if you are going to treat a patient for thrombosis or for extensive fibrin sheath development, it would be best to treat them prior to that 7 to 10 day period, because after that, when this becomes a more collagenous sheath, any thrombolytic will not be effective on removal. Now, having gone through all of that and having a clear understanding about how these infections develop, then we need to go back, understanding each step, and put in mechanisms to prevent them. All of those strategies involve preventing bacteria from getting into the portal of entry and developing a biofilm. This is going to require the compliant use of safe and effective technology. We are not going to be able to do this with process interventions, while they are very, very important, but to actually affect the bacteria and biofilm formation, it will require us to use technology. Now, this is something that I learned and understood a very long time ago, particularly in my practice as a nutrition support nurse specialist, where I was responsible for vascular access devices day in and day out. I learned about biofilm, and it was very clear to me this concept of using technology to eradicate biofilm infections or prevent them. And so I dedicated my career to working with industry to develop good programs of research and to develop effective technology so I could come back to you as a nurse, okay, and affect better outcomes for patients through good technology. So I'm giving you my disclosures and my work affiliations with industry. So back to our evidence. Well, and today we're going to talk primarily about the new technology with the chlorhexidine catheter. But what does the evidence, what does the CDC tell us we should be doing about this? Well, this is a Category 1A recommendation, and it recommends using an antimicrobial or an antibiotic coated catheter if the patient is expected to need it after 5 days. But going back to our data, most of the extraluminal infections occur in that 5-day period. So should we not be using it during that time as well? And here it says we should, unless our strategies are not working. And you'll notice that those strategies are primarily focused on the extraluminal surface. Now, this addresses the use of these catheters for the central venous insertion sites, but what about PICCs? And that's very important too. As we know, there's essentially no difference between the infection rate with CVCs and PICCs. So that is where we have this new breakthrough in technology in vascular access, and that is the introduction of the chlorhexidine-coated PICC. Now, we do have antiseptic catheters on the market, but one of the major problems with this device is that while it's coated on the inside and the outside of the body of the catheter, it does not have an intraluminal coating in the little extension tubing and in the catheter hub. And as we mentioned before, that catheter hub, and as the tubing extends downward, is the major source and major portal of entry for bacteria. So if this space is not protected, there could be considerable amount of biofilm develop in this area. So this is where the benefit of the chlorhexidine-coated PICC comes, in that the intraluminal surface, the extraluminal surface, the extension tubing, and the catheter hub are coated internally. Now, this is a graph of the data that was submitted by Teleflex to the FDA on the in vitro work that was done on a whole battery of organisms. This is a requirement for a 510(k) approval, and you see the results here of both the external and internal surface. Any device that's submitted for approval must have, with an antimicrobial coating, must have a minimum of a 4-log reduction. And you can see in this data that that was achieved in all bacteria categories. But the question becomes, how effective is this actually in vivo? So Teleflex approached me and asked me to study the efficacy of this catheter in an animal model. So I have now completed 5 studies to date on this catheter. Here are posters representative of 4 of them. The 5th one should be hopefully produced soon. Nonetheless, you can see in the summer of 2010, there were 2 studies done looking at the efficacy in the prevention of colonization of the internal and external lumen. In the summer of 2011, we went on to study the antithrombogenic effects of the coating as well as in the presence of bacteria. So we're going to talk about all 4 of these today, but in brief, and if you want to certainly look more into the methodology, the analysis, and results of these studies, these posters are available on the Teleflex website. So here is the first study, started in 2010. And I would like to introduce the research team that had these accomplishments. First is Dr. Robert Gunther, who is a professor emeritus from the University of California, Davis School of Medicine. He was the director of the Animal Research Laboratories there for about 30 years. Dr. Garth James is the medical director of the Center for Biofilm Engineering at the Medical Biofilm Laboratory, and Dr. Eleanor Pulcini is the supervisor of the students and technicians in the laboratory. Dr. Let's see, where are we here? Dr. Brezhnok and Dr. Sylvia are the veterinarians who were study directors of this study at the BioSurge Research Laboratory. Laura Bickle was the technician at the CBE who did all of the tissue analyses. And Dr. Al Parker was the biostatistician who did all of the analyses in the studies we're going to talk about today. So let's begin with the pilot studies in 2010. Two different studies, one looking at the extraluminal and the other the intraluminal surface. So our study question is there a difference in the microbial adherence or colonization to the intraluminal surface of a chlorhexidine-coated catheter compared to a non-coated catheter? All of these studies were approved by the IACOC, which is the Animal Subjects Committee, and is the equivalent to the IRB for human subject approval. All of the studies were experimental in design, and this study duration was 8 days. In the control group, we had 4 sheep with 4 catheters. Each sheep had a catheter in each jugular vein, and the same in the treatment group, 4 sheep, 4 catheters. Our methods, each sheep was randomized to a catheter group. The sheep were intubated and put under anesthesia. They had a preoperative skin cleansing with a chlorhexidine scrub, which is what I also recommend for your patients as a cleaning method. And then the sheep was taken to the operating room and a surgical prep was done with chlorhexidine and alcohol skin prep. We used maximum sterile barrier, and the catheter was inserted per manufacturer's instruction with the modified Seldinger technique. And the catheter tip location was verified in the distal SVC and right atrial junction with fluoroscopy. A neutral displacement connector was placed on the catheter after insertion and flushed with heparin 100 units per mil, and then we placed a chlorhexidine foam disc and vet wrap. Now here we're studying the intraluminal surface, so I'm using every intervention to prevent extraluminal infection. On day 1, after catheter insertion, we injected the catheter to fill volume with an inoculum of Staph aureus 10 to the 3rd per mil and was left there for 3 hours and withdrawn. On day 2 through day 8, we did post-insertion infusions. I scrubbed the connector with a scrubbing device, and then we gave an infusion of lactated ringers 50 mils per hour times 8 hours, and then again flushed the catheter with saline and heparin. The insertion site care was done every 3 days with the chlorhexidine alcohol skin prep and the foam disc and vet wrap. The connector was exchanged every 3 days, and paired blood cultures were done on day 0, 1, 2, 4, and 8. So what were the results? In the control group, 1 catheter was removed due to infection, and the other 3 made the full study protocol. In the treatment group, all 4 of the sheep made the entire study protocol. From a clinical assessment, we monitored them on a daily basis, and here are the temperatures of the in the treatment sheep. You'll notice that they are as high as 104 degrees, but 104 is the high normal for the sheep. So all of the treatment sheep remained afebrile. Comparing that to the temperatures in the control group, notice that we begin to see fever at about day 3. And again, the one that was removed was febrile on day 7. From the catheter blood cultures, day 0, as you recall, was the day of inoculation. So by day 1, I would expect to see some biofilm formation, and we did see it at a very low level in the treatment group. However, within 24 hours of that, the chlorhexidine coating eradicated all of the bacteria that were attached and remained negative through the rest of the study. Compare that then to the treatment sheep, okay, where we had a very high level biofilm formation up to 10 to the 5th in per mil in the control group. So this is actually a pretty good picture of what happens, you know, in the rapid production of biofilm in catheters that are unprotected. Now, when the samples, the sheep were taken out of the study, we did removed the catheters and sent them to the Center for Biofilm Engineering for analysis. When they arrived there, before any procedures were done, all samples were neutralized for the chlorhexidine. This was to prevent any false negative cultures. Looking at the biofilm formation, then we studied 3 areas: the catheter hub, the extension tubing, the catheter intraluminal surface at the vessel entry, and 5 centimeters of the catheter at the tip. Looking at the results, then you see in the treatment group that except for the hub, where there were 6 CFUs, all of the other cultures were negative. And compare that to the controlled group, where we see 1 and 2, almost 4 logs in the tip segment. These were statistically significantly different in 2 and almost reached significance in the other 2. So that was the intraluminal study, and again we saw very significant results in terms of the prevention of colonization on the intraluminal surface, including the extension tubing and the hub. So now we'll look at the extraluminal study. And again, the study was approved by the IACOG, and the study duration was 10 days. In this study, we had 4 sheep in the control, but we had 2 catheters, 1 in each jugular vein, and the same in the treatment group. The differences in our methods were that now I'm studying the extraluminal side rather than intraluminal. So I am not going to use any chlorhexidine products or any antiseptic for that matter because I want to know how protective this chlorhexidine surface is. So in the preoperative skin cleansing, cleanse the skin with soap and water, and then applied alcohol and allowed to dry to kill the normal skin flora. And then going to inoculate an area, 2 square centimeter area over the insertion site with Staph aureus 10 to the 6th. I chose 10 to the 6th because that is the normal colonization rate in the male of 10 to the 6th per square centimeter and 10 to the 5th in female. So I spread that out over 2 square centimeters, trying to represent a pore skin preparation. The catheters were then inserted through the inoculated skin, and a gauze dressing rather than a chlorhexidine foam disc and vet wrap were applied. Insertion site care was done with saline and gauze and vet wrap. The results then in the control group, unfortunately, 1 sheep was very bad, and he pulled his catheter out prior to the end of the study. 6 of the catheters were removed due to infection, and 1 catheter made it to the end of the study. In the treatment group, unfortunately, 2 of our samples were lost in shipping. This actually turned out to be a good thing that I'll share with you in a minute. 6 then made the full study protocol. So in total, we had 7 catheters in the control group and 6 in the treatment group. Looking clinically, again, at temperatures, which was our best indication of infection in the sheep, we once again see that all of the sheep in the treatment group were afebrile, where in the control group we see very high fevers early, and they began to show at about day 3. With the end block dissection then, here we see the skin, the catheter still in place, subcutaneous tissue, and the vessel itself all the way to the right atrium are taken out in one dissection. You can actually see the catheter within the vessel here, and that is because the vein walls are transparent. Looking at the tissues then, we excise the skin and then the subcu tissue. And here you see that collagen capsule that develops within the subcutaneous tissue around the catheter. At the vein entry site, you see the catheter going through the vessel, but one really remarkable thing here is you notice that there is no fibrin once it's past the vein wall. Then the entire catheter was, vein was excised, and we can observe that there is virtually no thrombosis in this vessel, nor is there any fibrin sheath development on this catheter. So let's take that and compare then to the treatment group. And here we see something profoundly different. We see a very high level infection, cellulitis, edema. And comparing the subcutaneous tissue in the control group, that is not a normal collagen capsule. That is very necrotic tissue and cellulitis. Looking at comparing the vein entry site, we see that once through the vessel wall, now we see fairly extensive fibrin sheath development. And then on the excision, compare the treatment group to the control, where we see a very high level of infection, cellulitis, a lot of clotting both in the vessel as well as around the catheter. This is a very perfect picture of septic thrombophlebitis. So looking then at the analysis and the biofilm formation, we looked at the tissues, the skin and the subcutaneous tissue. And from the skin, we see that we had a good 2 log reduction, but it wasn't 0. We still had 4 logs at the skin level. So this represents that it is protective, but not 100%. And in the sub-Q space, we see that it also was about a 1 log reduction. Again, protective, but not 100%. It turns out that these numbers were statistically significantly different. On the catheter surface itself, we also see significant reduction at the vein entry site in the catheter tip, but in the sub-Q space, we see a reduction as well, but not yet statistically significant. It's very difficult in a study like this with a sample size of 3 and 4 to achieve statistical significance. So these were quite remarkable results. Now, remember the 2 samples that were lost in shipping. That was very disturbing to me for a lot of reasons. One is animals died for no reason. 2, it's very expensive. Okay, and we lost a lot of time. So from that point out, I became the delivery person for the samples. And I was most happy in a way that happened, because when I took the samples to the Center for Biofilm Engineering and observed the dissection, I was totally amazed to discover that in the treatment catheters, there was no fibrin sheath, or virtually none, and there was no venous thrombosis, even in the face of high-level colonization compared to the control groups. So my thought was, is it possible that the chlorhexidine coating could have both antimicrobial and antithromogenic properties? So I was very encouraged by this and encouraged Teleflex to continue study looking at the potential antithrombogenic properties. So with that, we did conduct 2 studies last summer looking at antithrombogenic with and without colonization. So first we're going to look at the antithrombogenic study. We had 2 groups. One ended at 12 days and the other at 31 days. We looked at 3 different catheters, the chlorhexidine acetate catheter, the Teleflex catheter with no coating, and then a third or a second control of a catheter that has a biomimetic coating. This is intended to be antithrombogenic. There were 3 sheep in each group, each with 1 catheter, so 3 catheters per group. In this study, we did the en bloc dissection, as well as measured the fibrin sheath in length and weight to determine the antithrombogenic. We also did histology of the veins and the fibrin sheath. So let's look at 3 examples of each group in the thrombogenicity study. Here we see the test catheter, the chlorhexidine catheter, an example at 13 days with very minimal fibrin attachment. And compare this then to the 2 control catheters, one at 10 days and one at 14 days. Interestingly, in the control 1 catheter, the one in the middle, when I removed that fibrin sheath, I observed that underneath that sheath was attached to the vein wall. So you can see here a lot of injury to that vessel wall with the adherent fibrin sheath. In terms of numbers, we did see a reduction of thrombosis based upon the measurement of length and weight of the fibrin sheath. So these percent reductions are compared to the test catheter. So those are the 12-day results. And then in the 31 days results, you see that there was even greater reduction of length and weight of the fibrin sheaths compared to the test catheter. These were not statistically significantly different, but from the perspective of as much of a 90 and 89% in length and weight reduction, to me, is highly clinically significant. So then the question was, okay, so if we get a clinically significant reduction of thrombotic material attached to the catheter surface, what happens when we put bacteria there? Because we know that thrombosis does promote infection. And on this slide, you can see the mechanisms by which that occurs. And it's basically that the bacteria are able to attach to the fibrin and develop a biofilm. But we also know that infection promotes thrombosis. And it does it by various mechanisms. The effect of bacteria and endotoxin initiates the thrombogenic cascade, as well as the leukocyte activation is procoagulant. So that prompted the second or the fourth study then to look at antithrombogenicity in the presence of colonization. So for these methods, we used the same 2 controls and the same treatment catheter, this time 3 in the 2 control groups and 4 sheep and 4 catheters in the test group. Our methods were the same except for a couple things. Again, we did the soap and water and alcohol antisepsis, and this time inoculated the skin at a 2 square centimeter space, but only randomly selected right or left jugular vein. And then for outcome measures, the same as the previous study, but I also added insertion site cultures, and we did some more additional microscopy and histology. Now I'm going to be showing you some histological sections of these, but before I do that, we need to refresh our memory about the anatomy of the vein wall and then look at a histological section. Here we know that the inside lining of a vessel wall is made of a monolayer of endothelial cells, and endothelial cells are anticoagulant. Underneath the endothelium, which is 1 cell layer thick, is the tunica media, and the tunica media is primarily smooth muscle cells. Recall that these are the cells, along with the endothelium, that will eventually cover the fibrin sheath. And the third layer is the tunica adventitia, which is loose fibrinous connective tissue. We also have valves and veins, and these valves are covered with endothelium. Now, looking at a histology section, this is a normal vessel, and on the intraluminal surface, you see a lining of what we kind of see as little black dots, and those are the nuclei of the endothelium. Underneath that 1 cell layer is the tunica media, and then the more loose fibrinous connective tissue of the tunica adventitia, and then surrounding vessels are connective and adipose tissue. So, what were the results? In the control group, 5 of the sheep had to be removed from the study due to infection. 1 sheep, 1 catheter made it through the entire study, where all 4 of the treatment sheep made the entire study protocol. This is a graph the skin cultures. We're cutting short on time here, but so I won't go into it in detail. But what you can see in all sheep that there was colonization of the skin during their entire stay in the study period, except for 3 of the treatment group had no growth on termination. Now here's the growth pathology in comparative. Here's an example of a test catheter at 31 days. You'll notice that there is no thrombosis or clotting in the vessel wall. We only have a small amount of fibrin sheath about 1 centimeter long at the insertion site. Compare that then, let's look at the histological sections first. In this section, the cross-section is at the subcutaneous tract where the catheter enters into the vessel. And you see that the inside of the subcu tract, interestingly, is now lined with epithelium, which are ingrown skin cells from the surface. You also see a section of the vein there. And remember that we said this column is contiguous. So we see the vessel there attached to the subcu area, and it is lined with normal endothelium. We also see the connective tissue and dense fibroblastic tissue that is the normal inflammatory process. Going down the vein then to midsection and examining the vessel walls, we see an area there that looks like it has some bumps on the vessel wall. Well, this is what we call intimal hyperplasia. It's somewhat equivalent to a scar. And this is due to mechanical irritation of the vessel wall. In this case, the maximum amount of hyperplasia in the treatment group was about 25%. Now we look at the control 2 catheter. This sheep was taken out of the study at 7 days, and again looks very similar to what we saw the summer before in the colonized vessels. When we do the excision, now we see very extensive thrombosis and infection and necrotic tissue. Again, a perfect example of septic thrombophlebitis. In the cross-section of the vein and the subcutaneous tract, we now see some very different looking material. And this is very necrotic and very inflamed tissue. Looking at the vein wall and the fibrin that's attached within the lumen, we see transmural necrosis and necrotic inflammatory cells. If you look up to the right area here, these sections of darkened material there are actually biofilm colonies. This is quite remarkable finding because this has actually never been, any photos of this have never been observed in any publication. And here you see on a closer look, you can actually see the biofilm. You can see the bacteria embedded in their colonies within this fibrin sheath material. Moving on down the vessel then midway, looking at a cross-section of the vessel, again we see transmural necrosis. We also see the necrotic tissue of the connective tissue lining the vessel on the external perimeter with proteinaceous edema. Within the vessel, we see the section of the loose fibrin there, and we also see a valve. But if you look closely along this valve, you see a lot of blackened areas. And zooming in closer, we can see that this valve and vessel wall are actually lined with biofilm colonies. And again, a closer picture of the biofilm colonies. Now, we've seen biofilm now in the lining vessel walls, lining valves, and within a fibrin sheath. Here is the control 1 catheter. This sheep was taken out of the study at 5 days as clinically septic. And here's a cross-section of the subcutaneous tract. Notice on the intraluminal lining, you see a lot of blackened areas. And looking closer at that, this is very extensive biofilm colony formation completely surrounding the internal lining of the subcutaneous tract. So, how does that translate into CFUs and numbers? Well, if you look under the fibrin sheath, the fibrin sheaths were stripped from the catheters and then were homogenized and made into a liquid, and then were plated to understand exactly how much bacteria was in there. This is quite amazing in that the fibrin sheath, what little there was on the chlorhexidine catheter, literally only had 2.5 CFUs. Compare that to the 2 control catheter fibrin sheaths that had as much as 10 to the 9th and 10 to the 7th. That is a 1 with 9 zeros. That is, I don't even know, gazillion, billion, trillion, way more than that. But that is just phenomenal in terms of an bacterial inoculum. Then we combined the measurements of CFU per square centimeter on the catheter sections that were analyzed. And there we saw as much as 10 to the 4th on the control catheters, but literally 5.0 times 10 to the minus 1 represents 0. You cannot use zeros in statistics, so you have to put some value, but this represents a 0. So going back then to the Pennsylvania study as a point of interest, looking at the time to endpoint. Again, this is an extraluminal study, so if our data matches the human literature, then we find what we would expect to see here, that these, in the control group 1, the animals became septic within 5 to 7 days, and in the control group 2 were at 7 days and 1 at 31 days, and all of the chlorhexidine sheep were not septic and made the 31-day protocol. So we're a little bit over time here, but, and I'm going to skip through this quickly. This was the 1 sheep in the control group that made it for 31 days. And if you look up on the 3rd week of the skin cultures, you can see that there was a 2+ Staph aureus and a 1+ bacillus, but that was the only time the bacillus appeared. That was the, I'll go back here, this was the insertion site on the day of necropsy, and you can see that the insertion site of the catheter is not looking too healthy there. So looking at the numbers for that sheep, we discovered that there was a non-staph species that appeared in both the fibrin sheath as well as on the catheter surface. And when we looked at that more closely and looked at the plates, here's the plate of the fibrin sheath that has both Staph aureus and bacillus, and on the catheter surface itself also has mixed species, which were Staph aureus and bacillus. And you can see the numbers there at 10 to the 3rd in the sheath and 10 to the 4th in the catheter. While this sheep remained afebrile on day 31, with cultures like this, I don't think the sheep would have survived much longer. But this brings to the point of how important it is to do good catheter insertion site care during the entire dwell time. So here let's, in summary, and let's compare the chlorhexidine test catheter, where we see minimal to no fibrin sheath formation, virtually no venous thrombosis, comparing that to the control 1 and the control group. So the conclusion of our study was that the chlorhexidine PICC is effective as an antimicrobial and antithrombogenic catheter surface all at the same time. This is indeed a breakthrough in technology and is the first antimicrobial coating that is both antimicrobial and antithrombogenic. So this data from these studies was submitted to the FDA for antithrombogenic claims, and here are the claims that they permitted. 61% reduction in thrombus accumulation on catheter surfaces after 30 days. When challenged with infection, 92% reduction in thrombus accumulation on catheter surface after 30 days. Reduced thrombotic intraluminal occlusion and reduction in phlebitis, and an average of 72% less intimal hyperplasia after 30 days. So with that concludes my presentation. I will be happy to take any questions you may have.